here to discuss pros and cons, difficulties, challenges, etc. So for this reason, we have invited people from different angles that can give us a different opinion, and probably we can ask that you have micros here, so what we are going to ask you is first to introduce yourselves, each of you, and maybe we can start asking for, the people that are on the screen. So maybe Krista, would you like to introduce yourself to the audience?
Sure. First off, I'm sorry I can't be there. I hear it's a really good conference, so I'm really sad that I can't be part there, but I'm delighted to be part of this roundtable. So I'm a faculty at the University of Florida here, and I direct the Imaging NMD program.
Thank you very much. Paolo?
Similarly to Krista, I have tried to come there, but unfortunately I had a final call that kept me here. So I'm Paolo Bettica, I'm the Chief Medical Officer of Ital Pharmaco. And as you know, we have used MRI in our phase 3 study, the EPID study.
Okay, so maybe we can start now from there. Elena, would you like to introduce yourself?
Thank you very much for the invitation. My name is Elena. I work at EMA. I'm a neurologist by training. And before joining agency already 6 years ago, I worked as a clinician, also doing research on MRI, in particular for multiple sclerosis. Thank you so much.
Okay.
Yeah, also thank you for the invitation. My name is Erik Nix. I work as a neurologist in Leiden University Medical Center, involved in pediatric and adult clinical trials.
Right. Okay, so my name is Marcus Karlsson. I am a scientist at Amra Medical. We're a company in Sweden that works with MRI image analysis in clinical trials. So we've worked in neuromuscular diseases and trials of various phases and sizes, as well as in metabolic diseases.
I'm Hartman Reingoud. I work at the NMR laboratory at the Institute of Myology, more from the basic scientist part. We are very strongly involved in everything that's clinical trials, and that's that's my. Sorry.
Hello, I'm Kieran Hollingsworth. I am Associate Professor of MR Physics at Newcastle University. I have a long-standing both academic interest in quantitative MRI in terms of natural history studies, but also leading for both the running and application of quantitative MRI in clinical trials.
Hello, everybody. My name is Jared Sheehan. I'm coming from the medical affairs team in Sarepta Therapeutics. I'm by far the least expert of this panel, so I'll defer all technical questions to my right. But we have MRI across our studies of Duchenne, limb-girdle, FSD, DM1, and I'd like to share the insights from the biotechnology industry on some of the issues we might have and some of the solutions we might have, and hopefully have a great discussion up here too.
Okay, great. So how this is going to work? We have prepared some questions, but this is completely open to the audience. So we will start with questions and then we'll stop, and please just help us. So if you want to ask something, you are interested about a follow-up question, don't hesitate, just raise your hand and we will give you time for that. So the first question I think is for all of you, so you can volunteer to answer, but the idea is that after what we have discussed during these days, do you think that imaging without functional data can be already considered an independent outcome measure for monitoring on pharmacodynamic biomarker clinical trials? So you think that we can use imaging for clinical trials already as an outcome measure? Who wants to start? Kieran?
Okay, so I guess what do we see in the MRI measure? If we look across everything we've heard here, what keeps coming up is fat fraction, fat fraction, fat fraction, and we know from both the natural history studies we've got and are still being done, from the longitudinal some of the longitudinal therapy trials that have been done, whilst measuring fat fractions, that we're seeing a measurement that we're able to do in multiple centres, that we can relate to the progression of the disease, and in which we can sensitively detect change over reasonable periods of time. So that's the power of what we have. In the sense of the way that that clinical trials are defined, ultimately in a development course, you've got to have a clinically relevant outcome. But that outcome, those measures may not be sensitive enough to detect in a small period of time. So, in the interest of testing something and failing early or getting encouragement to carry on, having the most sensitive measure is, of immense value. And I think that's why we're seeing all the work on fat fraction. We're still seeing, a predominance of effort in that area.
Okay, Elena?
Yes, and as a follow-up, I do agree that I see a great potential for the MRI as a clinical outcome measure, but not as the primary endpoint for the confirmatory trials, but probably, yes, as a good method to rapidly scrutinize what is probably to work, because now what we see is that we measure certain proteins. Duchenne is probably one of the best examples. We measure the proteins, and they are very promising results, but then it doesn't translate into the functional benefit, and there might be valid reasons why this is happening, including potentially that the functional tests are not the best design ever, but it is also possible that the drug doesn't work. So in a way that having something that could be within this ballet to make a rapid decision on what programs should move ahead would be a very valuable idea. So I encourage the field to move forward into that direction.
Okay, I think Paolo wants to say something.
Yeah, maybe it would be interesting to remind us what we have done. We have used fat fraction with many different objectives. I mean, one was to pre-select a population that was at a defined risk of losing ambulation, because we know that actually one significant problem when running clinical trials looking at the effect of a drug in Duchenne is to select either a population that is not really progressing, or on the other hand, to select a population that is progressing too fast, and therefore at the end you end up with a significant number of patients that actually cannot really perform the test. So we have actually used as part of our inclusion-exclusion criteria a predefined level of fat infiltration to ensure that we could select the best population to test the effect of our drug. The second use was actually as a secondary objective, where actually we had a significant effect of givinostat on the fat infiltration. And this was actually very important in the, evaluation, a sort of holistic evaluation of the effect of givinostat in Duchenne. So to me, while certainly using fat fraction as an early assessment in a sort of proof of concept study is certainly of very important value, I think there is very significant value also in a pivotal trial, even if at the moment we are not maybe ready to declare it as a primary endpoint.
Okay, so I think there are more. Marcus, you wanted to say something?
Yeah, I was gonna sort of in our experience, we have worked on the studies where we've used like either fat fraction or lean muscle volume as a primary outcome, as an efficacy outcome in phase 2 studies. So I think that's workable, but as I think we all said, it's not ready to be used as a primary outcome of a clinical benefit in a phase 3 study yet.
Okay, and then you also wanted to say something, right?
Just one point. I think, you know, in other diseases, we see cardiac MRI being used as a primary objective. I think it's also important to look there, looking at ejection fraction. We have a really nice case where we see muscle, we see this change over time. It's replaced by fat and fibrosis. We can measure these things using MRI. So it makes completely sense as a poster child for this endeavor that this community is pursuing. I think one of the difficulties is the variability you see in these diseases, such as like one case with limb girdles. This is a very heterogeneous disease. You see it very slow at the start, and then you see it's progressive, obviously, later on, and this is where you see a huge variability. So actually to find these patients, these are ultra-rare diseases, to see these changes over time, it's very difficult to show a correlation with functional measures, and we know they are what we have, but they also lack this specificity that we're looking for too.
Okay, and Krista, I think you wanted to say something as well.
Yeah, I'm sorry, I can't find my hand raised, so I'm gonna raise my hand like this. I'm gonna agree with everything was said. I think that certainly the MR measures of fat fraction have really proven to be extremely valuable in the context of both natural history studies as well as Paula mentioned in clinical trials. I do think that obviously for it ultimately to be accepted as a surrogate endpoint, it has to show not only that it correlates with function, but it also predicts change in function. I think in Duchenne, I think there is a lot of evidence already that that is the case. I'm not sure if it's in all of the diseases yet, but I certainly think there are diseases where we have a lot of evidence that that is the case.
Elena?
I want to build perhaps on the case presented of using the MRI as an enrichment marker that was done indeed in the study of Dubinothat. Indeed, that could be also a very good idea, because today and yesterday we discussed this notion of not having there is clearly a non-linear relationship between the MRI pathological phenomena and the clinical expression. So if it's possible to understand this non-linear idea, to identify which are the study population that is prone to show the informative outcome in a short trial or relative short trial, that would be very also a good idea, because that's a very great limitation we have with long-lasting and slowly progressing conditions, and that's particularly also even more problematic for pediatric conditions. So that's another very good option.
Yeah, maybe to add to Kieran's original comment, I think, so I do agree that we see more and more about fat fraction, but I think we also have to realize that this is clearly within this community. And we had the excellent presentation yesterday that you also see where there was the initial increase in studies using the MR fat fraction. You also see a kind of stabilization or even a decline and an increasing number of protocols that do not include MRI. And I think the importance in the development is, of course, that you will need a substantial number of studies, different diseases, different stages, in order to build your case, in order to be as an endpoint, especially in the rarer diseases with the smaller groups. So I do see a risk there. And well, the problem is that if you don't have a consistent protocol throughout different numbers of diseases, then you will not build this case also for the rarer subtypes that will follow. And I think this is an important risk. So I do agree that we need drugs that work, and of course they need to show, the MR does also need to show response to treatment, but if it's not in the protocols, we will not know. And I think that's something we should realize. It's now up to individual companies whether they include the MR, and of course, as was said, fortunately, many studies do, but not all of them. And I think this is a risk.
Mm-hmm. Yes, and also commenting with that, and with a comment about the surrogacy. So the idea of having a surrogate somehow entails that you are able to provide evidence that the effect of the medicinal product on the surrogate translate to the clinical endpoint, and there is no off-label therapy impact. So in a way that the medicinal product does not affect the clinical outcome, if not through the surrogate. For this, the very minimum is to have an efficacious medicinal product, because then it's impossible to test. So that's why I think it's somehow very difficult to go into this direction, because then you need efficacious drugs. I do agree that if you don't include the MRI protocols, you will never have it. But then the first time someone has that level, then a question entails, well, for the second, why the regulatory agencies would ever agree on a less important evidence. So if one drug has proved to have a clinical effect, while the rest of the drugs should be based on a less, let's say, comprehensive evidence. That's always a question I have in my mind. I think there are good reasons for the pharmaceutical companies to argue that, well, you could save money to do the trial because you need to recruit less people and follow for less of a time. But at the same time, I always have this dilemma. If someone makes the case why the second one should be only based on the surrogacy.
So if I can answer that, so my idea was that it would be, of course you don't want to lower the bar per se, but I think there will be subtypes and specific diseases where you just will not have the numbers to statistically prove clinical difference. And there I think it's not about lowering the bar, it's just that you need a different marker in the end, because your clinics will not have the numbers.
So just asking a question, about what you've just said in terms of how you regard the different, the direct functional measures. I mean, is it taken into account the fact that many of them are subjective? Can you take into account the fact that there's variability in motivation and effort as opposed to something which is independent of those things? Because I think that stands as quite a core reason why the MR is developed, why the MR might be useful. I can see if you insist on direct function, that is one thing, but then you've got to recognise that you are either running larger and larger trials or longer and longer trials, and you might not want to. That may not be your first or intermediate step.
Yeah, not only that's the case, for sure the variability of the functional test is likely higher than the variability of the MRI, even if yesterday we heard many sources of variability. But I tend to agree that that's the case. And there is also the impact, as we know, about potential external factor impacting how well are you doing the 6-minute walking distance today versus tomorrow. That's been said, and as has been recognized today, to authorize a drug, you need to have an effect on the functioning of the patient. So ultimately, you will have to show this to some extent. There are other conditions that face the same exact situation as is happening with some of these conditions discussed today. One of the best examples is perhaps Alzheimer's disease. So there, they can also argue that the accumulation of certain proteins is 10, 15 years before of the appearance of the first symptoms, which is the case. Yet they managed to study the condition so that they can identify how these curves of damage correlates with the functional or the changes in the cognition, which are also very variable, and determine which is exactly the exact population they should study. to do the trial, because also they recognize it's not possible to do an Alzheimer's disease trial lasting for 5-10 years. That is not only very expensive, you're going to handle a lot of problems with the missing data and the quality, and it's not going to work for anyone. So they solve that situation with this very accurate understanding of how all of these dynamics are interconnected. I don't know whether it's possible or doable to do for neuromuscular conditions, of some of them, at least I hope it is.
So maybe we'll move on. That was a great discussion, and we'll move on to the next question. So for our academic and colleagues from companies, what are the biggest difficulties that you face to include imaging in your studies? And maybe some of the things you've done to overcome them as well.
I can start off just a few small things. So I think one of the key issues is, you have to go to many sites across the world to actually do these studies. You need to make sure that you have harmonization across the protocols. You need to make sure technicians are on the same page. We're collecting data in the right way. Of course, patients need to be able to be getting into this machine. I don't think anybody wants to be sitting in here for an hour in an MRI, but to make it as stressless as possible, to make it them comfortable. This obviously is very difficult in Duchenne when they're non-ambulant, and you have to get them into the machine too. And this is a constant issue, I think, for anybody implementing MRI. It is an interesting approach with, for example, external comparator group data. Like this is something that after you have a placebo-controlled study for just say 1 year, you want to follow these patients up for multiple years, and it's important to have some data to benchmark against your treatment to say, what is this fat fraction over time? But this data is kind of very difficult to get to minimize this bias, and I think there's obviously multiple efforts in order to do this, but this is something I think that's missing from the community level to pull all this together. Maybe I'll just stop there. I'm sure there's others who want to share.
Yeah, Paolo?
Yeah, I think with regards to the development of drugs and clinical trials, standardization is probably the most important part, and I think there is a trade-off here, because obviously if one is applying a very standardized approach, maybe one would be not using the very latest MRI technique or method. So there is a trade-off that we need to find. But I think in a clinical trial, standardization is probably the most important part. And in fact, we have spent quite a lot of effort in that regard. I think thinking about the future, it would be very important to understand how relevant is the standardization in the acquisition. Because in the EPID study, we actually had a limited number of MRI sites where actually patients had to go to, obviously putting a very significant effort. If that is something that we could avoid having, if you're on the MRI site and the clinical site being in the same hospital, that I think would be already a significant improvement.
Okay, then just a follow-up question probably for Kieran, Harman, or Marcus, or Krista. You have helped sites to standardize the methods. So what are the difficulties that you face when you try to do that?
Yeah. So just a couple of comments first. So I definitely agree with what was said. Standardization is really key. Standardization and consistent handling of the data, which also acquires, I think, in-house and centralized data processing is really important. I think the challenge still comes down to not every setting is set up to deal with research studies and follow standardized protocols. I think it's sometimes hard in certain settings. Clinics, as you know, that use MR facilities are very busy. Not all of the technicians are necessarily going to be ready to follow your protocols. And to be quite honest, a lot of time, it still comes down to effective communication, data export, and making sure the data come to us in a standardized manner. So the simpler the methodology, the more likely you can standardize. So we have been using the same sequences for the last 15 years across the natural history studies and all of our clinical trials to help build that large database, but also so that the sites can run the same protocols and it's as easy as possible. In our case, we've been using proton spectroscopy, single voxel, very easy. And the other strategy that we've used is actually where we run 2 sets of 4 gradient echoes to do the Dixon in-house reconstruction. So the simpler, the better, I think.
So I think in terms of, in terms of that wish, how do you, the difficulty of having the sites that you want. I think what is more encouraging now, as Krista said, absolutely true. When you look at some sites are just not suitable for doing quantitative research, and that you can find out through things like careful questionnaires about how many operators they have, what time they have dedicated to research. Ideally, you want your site to already have a good CV of quantitative research. But I think what is encouraging when I look at the clinical trials that we're involved in different capacities is that now there's a larger number of networks of sites. I was just having a brief count through, and I counted 7 distinct networks of sites where quantitative muscle MR is going on in a standardized and qualified manner. And, some of those networks certainly have been expanding rapidly, in recent years. So no, you can't bring every site on board, and if you want to bring a new site on board that doesn't have that CV, you have got to spend both money and time to do that after very careful vetting. But I think the good news is that there are already pre-existing networks which do exist to do that. You may not be able to scan right at your intervention centre, but hopefully not too far away.
I mean, and I think in our experience, I think as people have said, the important thing is to keep all your imaging as simple as possible. So we typically work with very, very basic, just 2-point exon sequences and try to implement it in a way so which really should really just be almost like a push button exercise for the MRI techs. So it's really easy to not screw it up, and then also sort of having a good processes for monitoring data and catch errors, and then and sort of work on how do you send feedback to the sites and so on, and also really, I think, keep in mind when you design your whole system and process that you want it to sort of be able to run it in a site that is not a sort of specialized neuromuscular research radiology center. Like get something simple that you can run in other settings as well.
Yeah, very difficult to add anything to that because they're very valid points. I want to make some additional points. Is that there was the mention or the issue that it would be better to have the clinical site, or where the PI is, the neurologist, and the radiologist department is in the same hospital. That's of course the ideal situation. That's also what we recommend as a central reader. The reality is, of course, and there are some industrial partners, is that sometimes they have this pressure that they need to use the radiological department there where you have the PI. So we have the main word here is that you have, as a central reader, you have to have adaptability. So of course simple protocols are very important, but every site is different. The people you need to get around the table, I think, would be the vendors, but not together, because probably they won't want to do that, but separately, at least. That would be a good thing. And the MRI technologists, maybe more than the radiologists, are very important in this. don't think they're represented today, but they are doing the work. There was a vendor or an industrial partner that had a good idea, is to put all these MRI technologies into a forum, because they can help each other probably. Because most of these MRI technologies, they do most of the time brain, if they're in a clinical MRI department. So sometimes they have to do a muscle, and if then it's quantitative muscle, then it's more complicated. And if they do it once in a year, it's better than there's some kind of network. I don't think it exists. It's probably very difficult to set it up, but it's a good idea at the very least.
I was still trying to remember your original question, but I think it was about limitations, right, for companies to include MRI. And I think the problem here with this question is that you have an audience of people who are strongly in favor of MRI, and also companies here present who do believe in the technique. And so if you talk to companies who have a different approach, I think what you, of course, hear is its costs. It's quite troublesome to include. They want to include as many centers and go as fast as possible. Adding MRI, which of course give more difficulty with the implementation. You have to select centers, you have to train centers, and so on and so forth. So I think that's a problem. I think they also are not helped by the fact that, in my view, we could probably even also benefit from either some more guidance or even more some urgency from the regulators, how they perceive this biomarker. So this is debate of how valuable is it in the end. So that for me would also be a question for maybe Elena or for Krista. So if the regulators could somehow be even more proactive and provide more guidance than they have done in the past years.
Yeah. Krista?
I was still responding to a comment earlier, so but I agree with what Eric said. I think that more guidance from the regulators would be really helpful, and more support from that angle. The comment I wanted to make is really related to standardization again and the training of the MR technicians. One of the things that I would like to see is that the vendors, that the sponsors include the MR team in their investigators meeting. I think in the same way as we've done for years for functional evaluators. I think that would really create a network of MR technicians that are really capable. And the same way as we've had to do it for physical therapists, train them and really make sure that they know all the components, and they're also invested in the study. So that's one angle that I would like to present to some of the pharmaceutical companies, to include the MR team in their investigators meeting, which I know certainly Pharmaca has done that very effectively.
Elena?
Yes, and I have also a comment regarding your original question. I see also another risk depending on the complexity of the protocol. If there are few centers capable to conduct that protocol, there is a potential of a problem of generalizability of the results of a trial. And that obviously is a trade-off. So the community may need to decide on whether maybe it's a less fancy protocol, but doable for a large number of centers might be preferable for certain scenarios. Again, and perhaps connecting with your question, I don't think you will find a position about MRI, because at least for us in EMA, we qualify something on a context of use. It implies what is the use of that, I don't know, the percentage of fat conversion in that condition for that particular use in this kind of trial. So it's not going to be a position, global position on the use on MRI of neuromuscular disorders. I don't think this will be ever achieved. So that would be my comment regarding the limitation. So that there is, I see that potential.
So for the audience, we just want to open it up to see if there's any questions. We can keep going with our questions, but we'd love to hear if there are questions from the audience for our esteemed panel.
No. Yeah, so as we've seen a lot today, and I couldn't make it yesterday, but I assume yesterday as well. There's been a lot of talk on fat fraction, like Kieran said. And just because I'm an early career researcher, you guys have been talking about the importance of simplicity for multiple, and that makes perfect sense. But just the sort of thought that I come with is that at Newcastle, where I work with Kieran, until we had a new scanner installed recently, the old one, to do Dixon, we actually needed a patch. So at one point, wasn't Dixon complicated? So where is the trade-off between innovation and simplicity? Because fat fraction, sure, we can see it tells us a lot, but there could also be other things, as we see possibly with T2, where we're looking at a different stage of the neuromuscular disease process, the actual activity, rather than the damage done. Maybe I'm completely misinterpreting it, but it seems almost like the conversation is discouraging us from looking at doing new things just for simplicity's sake.
Yeah, I guess it's a really interesting point because, once upon a time, back when I started looking at this, everything was hand done, and everything was everything had to be completely run, run yourself. And I guess the question is, you look at the different modalities, and you have to ask what do they do? Back in the day, Dixon was investigated as a new kid on the block. Really the in and outer phase, found its early clinical application in really qualitative liver imaging, and then started to be applied to other areas, and then muscle became a very important one. I still get, of all the people who come to me for MR and queries and all the exciting things I want to do, the vast majority of them, whether they're neuromuscular, whether they are people looking at the oldest old, arthritis, sarcopenia, blah, 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 the most common query effectively is still Dixon related in some way, or it's fat water related in some way. And, then the question is, what should we explore scientifically? What translates then into being a good thing in a clinical trial? So for example, sort of being provocative in muscle, we see a lot of good scientific work about diffusion tensor imaging in muscle. Is it a suitable thing to cross into a clinical trial in muscle? Not really, because of the number of complications and complexity that underlies it, both in acquisition and reconstruction. But, there's been an academic phase of development of Dixon for fat fraction, for the interpretation of water T2 from different sequences, and both of those things have got to a point at which we're able to say, there is intrinsic value to those things. But it doesn't mean that we should stop looking scientifically at other things and keep pushing that case. So we've got to keep in mind our two hats on. We don't not do it just because it's complex now. It's about demonstrating the value of it and making it happen. Once upon a time, the MR vendors wouldn't get on board with Dixon for fatty liver, and nowadays they've all jumped on that ship. Okay. So, it's about finding what is useful, what is informative, and then pushing that very difficult agenda.
Just to build up on this, so it's a matter of whether you are working for the today or for the tomorrow. So someone has to work for the today to build up a comprehensive and compelling case for perhaps the simple measurements, but also another part of the community, or maybe the same community, in another time need to work for the future. So it's not, that would be my honest answer, is what are you looking for, actually?
Another one of your colleagues yesterday, Andrew Blemeyer, said that it took 25 years for the Dixon to be picked up. So I think that already answers a little bit the question. So it's something as simple as that. It took so long. So people are slow, unfortunately. There's a big role for the vendors again. We mentioned it many times. Why can we make it work on some sites, and why is it so difficult to then implement it in sites that use more or less the same scanners? So there's something that we need to really tackle there. I'm always in between discussions with someone from the vendor and then the site. At some point, is it a loop? Why is it always some kind of? So I think there is some way to break that vicious cycle and to make it work. I don't know. Maybe we need more data. I don't know why. It's muscle. It's neuromuscular, so it's rare. So that does not help. There are very, very few radiologists doing neuromuscular. It's more neurologists that are in the field, and then you have the MRI technologists. The radiologists are very rarely in. There are very few. So that's also a part of that. But I think if we can get the vendors to be a little bit, to have some kind of community within hubs. Kieran said that there are 7, that he distinguished 7 sort of networks. That might be the basis, probably in Europe, or it's also in the States. That might be the basis of some kind of discussion with the vendors. Per vendor, then.
So, Krista, you want to say something else?
Yeah, I think that the point that was made by the junior investigator, I think is a really important one. I think it can be really challenging to balance that. Clearly, when you're doing clinical trials, you're trying to do standardized sequence. It has to be simple. Same thing for natural history. So we always try to keep 2 arms going for our program. On one hand, we have the clinical trials, which are much more standardized. They have to be repeat. And then the other arm is really where we focus on development, where you try new techniques, you try pushing the envelope. And at some point in the future, that technique may be ready to move over to the clinical trials. But I think it's really important for the junior investigators to realize that. And in fact, a lot of times within our setting, we try not to have many of the junior investigators involved on the clinical trial side, because there are some risks associated with that. And I just, wanted to make sure that that message was clear for some of our junior colleagues.
Volker?
Yeah, I think a lot of this has already been said, and Kieran just pointed this out as well. The discussion is extremely helpful, and I don't think it's about, will MR endpoints be approved by regulators, yes or no? At some stage, I'm certain there will be. And we have seen this in the non-alcoholic fatty liver diseases. We have seen this in multiple sclerosis, where it took years also, before this became an accepted endpoint. Elena and I had a brief discussion. Most of the endpoints that we use for drug development are surrogate markers, whether it's blood pressure, whether it's cholesterol level, whether, it's other biomarkers as well. So I think it's still early days in the neuromuscular field. We do not have that many drugs that show a strong effect size, where we can show clear functional benefit, that have also used MRI. And the Italfarmaco trial is one of the exceptions, really. So yes, I think when we have more data in effective drugs, and we can show superiority, a better predictability of MR. And I think in some of the talks also today, we make the mistake by often just referring to correlation. If MR correlates with clinical function, why use MR? Okay, it needs to be having a stronger predictability. It needs to be superior in its outcome. It needs to allow us to power trials better. So, but this is us as a community to show to the regulators, of course, to collect these data. And hopefully the conference, contributes to making a case, convincing companies, academic trialists as well, to use MR.
Eric?
Maybe one short addition to the discussion about the hard work on the simple measurements and the maybe more exciting work on new new markers. I think, so what already has helped a lot, and I think hopefully many of the very brilliant young scientists can help us more, is by shortening all the protocols. So finding very clever ways to limit the duration of the protocols. I think in general you could say that for a pediatric protocol it would really be up to 30 minutes, ideally much less. Adults maybe with 1 hour if you're lucky, but also there shortening I think would indeed give much more possibilities. And I think if that would evolve, and hopefully would evolve much further, I think you can indeed work on today and while at the same time do some exciting stuff for tomorrow. Because I do agree that we need more, not only, yesterday we had a discussion that fat fraction is of course important, we have a lot of data, but in the end it's end-stage pathology. It will likely not change with either compound that you test. So similar to multiple sclerosis, more markers that reflect disease activity, or whatever that may be, I think would be highly needed. Yeah.
And just connected with that, I mean, even if I still believe will not be a primary endpoint for a confirmatory trial, there is still a value to have this MRI as a key or secondary endpoint, not only to support the functional, but actually, if the field is managed to get an MRI marker that connects with pathology, knowing that there is certain degree of difference, but for me it's relevant to substantiate the disease-modifying claim, and also if it happens that the trial is not ultimately truly comprehensive for a full marketing authorization, the fact that you compile evidence from different aspects in a reasonable manner to build a case helps, and that I think is what it helped actually for Italfarmaco for having the conditional marketing authorization, and it has helped other developments. And a recent example has been Qalsody for SOD ALS. That it's very obvious that the primary trial failed the endpoint. That's a fact. Yet the totality of the evidence, including the evidence on neurofilaments, help to build the case. So there is also that, for me, that clear to go into MRI endpoints that actually connect with the pathological course of the condition.
So for our next question, how can we use imaging for post-marketing evaluation of the drugs? Shall we use the same imaging protocol that was used in the clinical trials?
It ultimately depends on how is the use you're thinking about MRI. If it's for example, I don't know, enrichment, as it has been said, or even stratification, maybe you don't need an extensive protocol, ultimately. If you are using the MRI to substantiate a clinical endpoint, maybe you need a more extensive. There is always the opportunity to try to include other potentially promising markers to try to move ahead with the validation process. So it depends on how you feel. I mean, probably a certain degree of adjustment is needed. We've seen this with the clinical outcome. The fact that the clinical outcome is a good one to measure the progression in the clinic does not necessarily mean it's a good one for the clinical trial. So probably it needs some sort of deliberation on a trade-off what to include.
Okay, so but what the other thing? Do you see MRI as something that we can use once the drug is approved, for example? We have been talking about the challenges of standardizing, etc. But now the drug is approved and you want to follow up your patients. Would you use MRI as has been done during the trial to follow up them? What do you think, Eric?
I find it, to be honest, very difficult post-marketing. I think if the drug is approved, it's about access, and patients want access to the drug and as soon as easily as possible. So then you get into the issue that you want it maybe not via the expert centers, but you want it via a different center. So you run into the problem that including MRI in such a thing, besides it would be very, very costly, you would again run into the problem of standardization and can each center do that? And I think so we have some experience now in adding MR protocols to the standardized follow-up of patients. It's a challenge. It's a challenge both with regard to the burden, because it's a long day and they have to do multiple assessments, see multiple physicists, of multiple clinicians, and adding MRI is difficult. So I find it more challenging even than trials. Yeah.
I mean, I think I agree, because because in the trials you will have everything sort of as standardized, harmonized as possible. You will have a central reading, central processing. So you would need to have something, maybe not the exact same thing, but at least more sort of commercially available ways of sort of processing and collecting quantitative images. And I don't think that's present today, but that's, yeah.
I think Elena wants to.
What would be the reason for that? Because just to put an example, when I was a former MS specialist, we do use the MRI for the monitoring, because if we see activity, then we change the therapy because it's not working. But if there is no other therapy, then what would be the reason?
Klaus, you want to say something else?
One other question. What is the role of spectroscopy in clinical trials? Is there a role? We know that the precision is typically not as good as Dixon.
Okay, who wants to take that one? Krista and, well, Kieran, you had the mic.
Well, I guess, you know, I mean, I think for spectroscopy versus imaging, you have, you have a different balance of advantage, you know. So for spectroscopy, you need to be able to get the voxel in a consistent place. It requires you to know where you want to put that voxel in order to make the measurement that you want to measure. But we do know that we do know some of that information from the natural history studies that have been done. It is very much about the pre-information that you already have, and so for example, the measurement of water T2 is in many ways simplified by doing it spectroscopically as opposed to imaging. So it very much depends on the condition and what your state of pre-knowledge is.
Krista.
Yeah, so we have used both spectroscopy as well as Dixon imaging. I agree with Kieran that spectroscopy is really helpful to measure certainly water T2 because it's not as influenced by the fat content, which can be really challenging when you're dealing in more progressed disease states. It's also been very valuable in the context of measuring fat fraction. It provides a high signal-to-noise measure, which is really important, and particularly in younger patients when fat fraction levels may be low and you really need that sensitivity. The downside is that you need to select a voxel, and the MR technician selects that voxel in a particular region, where the advantage of the imaging approach is, of course, that you can look at multiple muscles simultaneously and you can make post-hoc decisions. So I think there's room for both techniques in the context of clinical trials, depending on what you're trying to do.
Any other questions from the audience? So that, I think we'd want to thank our panel very much for that very stimulating discussion.
So, and with this, we are closing the day. So before you leave, a few housekeeping things. First is that we are going to take a picture now. So if you don't mind, while you go out, go to the stairs where you are going down and stay there, because we are going to take a picture in the stairs. And after that, as you know, there is the reception, so there are going to be some drinks. So thanks again to all the participants today. Thank you, everybody. I see you tomorrow.
Thank you. Bye-bye.
Thank